Hello everyone, welcome to today's lecture. So we have finished with 4 chapters, okay? Chapter 1, Chapter 2, Chapter 6 and Chapter 7. Now we are moving on to Chapter 8, that is useful and harmful microbes. Now what do you mean by microbes? Microbes means microorganisms. This is a chapter which you have to read and study, okay? So you may keep your textbooks open for this. Okay, I will be reading out from the textbook and then explaining. First, can you recall what is meant by microbes? What are their characteristics? Microbes are unicellular microscopic organisms. Generally, they are prokaryotic. Fungi is eukaryotic. Okay, so it could be prokaryotic and eukaryotic. So you can write that microorganisms are microscopic organisms. How do you observe microbes? How do you observe microbes? With the help of microscopes. You are familiar with different types of microbes which are all around us but cannot be seen with our eyes. In what way they are related to our daily life? Okay, how we are related to them? We use certain microbes in our food preparations. For example, fermentation, for making bread, for making shrikhand, for making idli, dosa, yogurt, curd, cheese, etc., butter, or for making all these things we make use of microorganisms. Now the first microorganism which we have to study under useful microorganisms. In this chapter there are two topics, useful microorganisms and harmful microorganisms. First we will see useful microorganisms, then we will see harmful microorganisms. Under useful microorganisms, the first one which we are having is Lactobacilli. Okay, Lactobacilli is present in milk, it is present in curd. Now how it is observed? It is stained with methylene blue. You have to take a smear of curd. Now what do you mean by a smear? You have to take a slide, you have to put a drop on the slide and you have to spread it. Okay, when it dries, it is said to be a smear or a smear. Okay, I repeat, you have to take a glass slide, you have to take a drop of curd or milk on it, you have to spread it and it is allowed to dry. After it is allowed to dry, it is called as smear. Okay, on that smear you add stain. Okay, a stain or a dye. Here the stain used is methylene blue. When you see it under a microscope, okay, they have given the magnifications of about 10x objective and 60x objective. Now what do you mean by 10x objective? In a 10x objective, it is seen about 150 times bigger or 100 times bigger. In 60x objective, it is seen 600 times bigger. Okay, it depends upon the eyepiece of the microscope also. But for the time being, you may underline 10x objective and 60x objective. What do you mean by objective? It is the lens of the microscope. When you observe them under the microscope, you can see them as rod shaped organisms, blue colored, because the stain which we have used is methylene blue. Okay, so I'll read out the next paragraph. Did you notice the blue rod shaped organisms moving around? They are Lactobacilli, a kind of bacteria. They are minute and rectangular in shape. Lactobacilli are anaerobic. Anaerobic means which do not need oxygen. Lactobacilli are anaerobic bacteria, that is, they can produce energy without the use of oxygen. Can you tell how is yogurt made from milk? What exactly happens in this process? This is an important question. The next paragraph, till milk changes into yogurt. The answer is till milk changes into yogurt. So what happens? The lactobacilli which is present in the milk, it converts the sugar lactose which is present in the milk into lactic acid. I repeat, lactobacilli converts lactose into lactic acid. Where is lactobacilli? In the curd itself. Where is lactose? In the milk itself. Okay, so the lactobacilli converts lactose into lactic acid. As the lactic acid is formed, it reduces the pH of the milk. As the pH is reduced, the proteins in the milk, they get coagulated. Coagulated means they collect together, or they come together, or they precipitate together. They collect together. Asa gatta hoto. Okay, they collect together. The proteins in the milk, they collect together. That is said to be coagulation of proteins. Okay, that is how curd is formed. So we will see. The lactobacilli convert lactose, the sugar in the milk, into lactic acid. This process is called fermentation. As a result, the pH of milk decreases, causing coagulation of milk proteins. Thus, milk proteins are separated from other constituents of milk. This is what happens when the milk changes into yogurt. Till here, you have to write. Yogurt has a specific sour taste due to lactic acid. The low pH destroys harmful microbes present in the milk. Okay. Now we may move on to the next topic, that is use your brain power, page number 88. The answers are there in the notes. You may refer to the notes and write the answers, but we will just discuss the questions now. Why do doctors advise you to have yogurt or buttermilk if you have indigestion or abdominal discomfort? You might have noticed that at your homes also your mother makes you to drink buttermilk. Buttermilk means tak. Okay, tak प्यायला सांगतात. Why? Because it has useful microorganisms in it, and those useful microorganisms can kill the harmful microorganisms in the stomach. Okay, so that is why buttermilk are prescribed. Now the answer is useful bacteria in it help in proper digestion of the food. Next, sometimes yogurt becomes bitter and froths up. Why does this happen? Froths up म्हणजे फेस येणे, त्याच्यामध्ये बुडबुडे येतात. Okay, bubbles are formed in the milk, in the curd or the yogurt. Why? Due to excess fermentation, due to excess conversion of lactose to lactic acid. Okay, so the conversion of lactose to lactic acid is a fermentation reaction. Why it is a fermentation reaction? Because it is taking place anaerobically. Okay, anaerobically means in absence of oxygen. So the reaction which is taking place in absence of oxygen, that is roughly termed as fermentation. So sometimes yogurt becomes bitter and froths up. Why does this happen? It is due to over fermentation and excess of lactic acid production. Large amount of lactic acid is produced, and that is why it frothens up. Which different milk products are obtained at home by fermentation of the cream from the milk? Okay, different food products like paneer, cheese, yogurt, etc. could be produced. You may refer to the notes for the answers. Okay, now do you know, towards the right hand side, this is an important 2-mark question. What is meant by probiotic yogurt and other foodstuffs that are popular nowadays? Now probiotic drink, I will tell you an example. You might have seen the advertisement of Yakult. Okay, Yakult, Y-A-K-U-L-T, Yakult. Okay, Yakult is a probiotic drink. Probiotic drink means it contains useful microorganisms which will help in digestion of food in our digestive system. Okay, I repeat, probiotic drinks contain useful microorganisms which will kill the harmful microorganisms and improve digestion in our stomach. Okay, so I'll read out. Useful microbes like lactobacilli are added to these eatables. Such eatables are healthy because they kill the harmful bacteria like Clostridium in the alimentary canal, that is the digestive system, and help to improve our immunity. Okay, now the next topic, that is uses of lactobacilli. Various milk products like yogurt, buttermilk, ghee, cheese, shrikhand, etc. can be obtained by fermentation of milk. We know that. Lactobacilli fermentation is useful for large-scale production of cider. Cider is a fluid, it is a liquid. Apple cider means the syrup, you can say syrup. Okay, it is like syrup. Cider, cocoa pickles of vegetables, etc. Lactobacilli and some other useful microbes taken together are used to treat abdominal discomfort. Leavened fodder. Okay, fodder means chara. Okay, it is the food of cattle, the food we give for cattle. Leavened fodder offered to domestic cattle like cows and buffaloes is fodder fermented with the help of lactobacilli. It is a fodder which is made up of lactobacilli and oil seeds, the remains of oil seeds, and so okay, fodder means chara. It is prepared at in the evening and it is kept overnight for fermentation and it is given to the cows on the next day. Okay, the lactobacilli fermentation process is used to make wine and some types of bread. Okay, so there are 5 uses of lactobacilli given. Minimum 4 uses you have to learn. Okay, so that is the next question, uses of lactobacilli. Now research, next question. How many different industries depend upon the lactobacilli bacteria? There are different industries but mainly food and dairy. Okay, food and dairy industries. Which type of cottage industries and factories can be started in areas with abundant milk production? Okay, cottage industries and factories like infant milk food. Infant means babies, like Cerelac, Farex, you might have heard. These are milk food for infants, for babies. Cheese, Shrikhand, all these factories could be started in areas with abundant milk production. Okay, so this is about lactobacilli. So what are the points which you have to mark under this? It is the stain which is used, that is methylene blue. It is anaerobic. Anaerobic means which functions in absence of oxygen. Making of yogurt, the steps, it is a 3-mark question. Use your brain power, there are 3 questions, they are also asked. Do you know it is about probiotics? Uses, you have to learn 4 uses. And research, that is about the different types of industries. This much you may mark in your textbook now. Now the next organism is Rhizobium. You might have heard about the bacteria Rhizobium, about leguminous plants, okay, which live in the root nodules of leguminous plants. I will tell you what are leguminous plants. First we will start. Rhizobium, it is a symbiotic bacteria. Symbiotic means it is living in symbiosis with plants. Symbiosis means it is an association. Association means they are living together and helping each other. 2 of them are living together and helping each other. So there is a mutual association and it is called as symbiosis. So it is a symbiotic bacteria. Why? Because it is a bacteria which is growing in the root nodules of leguminous plants. Take a plantlet of fenugreek. Fenugreek means methi, groundnut or any other bean, not Mr. Bean. Okay, and sterilize it with a 3 to 5% solution of hydrogen peroxide. Sterilize means you have to kill all the microorganisms. If you apply hydrogen peroxide to the surface of those nodules, then all the microorganisms will die. Afterwards, keep it in a 70% solution of ethyl alcohol for 4 to 5 minutes. Again, that is also to sterilize it. Clean the roots with sterile water. Sterile means which do not have any type of microorganisms, that is distilled water. And take thin sections of the root nodules. Select good section and place it in a solution of safranin to 2 to 3 minutes. Okay, so what is the stain used? The stain used is safranin. Place the stained section on a glass slide, cover it with a cover slip and observe it under a compound microscope. The pinkish rod-shaped organisms are the Rhizobium bacteria. Okay, so they appear to be pinkish because the safranin is pink in color. Okay, and they are rod-shaped. Even Lactobacilli is rod-shaped. Note that we had to search for the root nodules of leguminous plants to obtain this bacteria. Are the Rhizobium bacilli useful to these plants or harmful? They are useful. Now I'll show you what do you mean by leguminous plants. Okay, so there is a plant, okay, in the roots of these, consider these are the roots. Okay, on the roots of those plants there are small nodules like this. Okay, like this there are small nodules. Okay, there are nodules. The Rhizobium bacteria live in these nodules. Okay, so the Rhizobium bacteria live inside the plant. Inside the plant means inside those nodules. Now what do these Rhizobium do? Okay, what do? They actually convert atmospheric nitrogen to nitrites and nitrates. We studied about nitrogen cycle, do you remember? So in nitrogen cycle, nitrogen is present in the atmosphere. It is about 78% of nitrogen, but that nitrogen is not used to plants, not of use to plants directly. Okay, it has to get converted from nitrogen to nitrites and nitrates, that is nitrogen compounds. So who converts it from nitrogen to nitrites and nitrates? This Rhizobium converts atmospheric nitrogen from nitrogen to nitrites and nitrates and make it available to the plants. Now why do plants need nitrogen? Plants need nitrogen to make proteins. Okay, proteins are not available in air, proteins are not available in soil. It has to be produced in the plant body itself. Similarly, when we eat the plants, we get plant proteins, but plant proteins is not of any use to us. When we eat, the plant proteins go to our digestive system. Our digestive system break those proteins into amino acids, and with those amino acids our body makes our proteins. Okay, so our proteins are different, the different animals proteins are different, the plant proteins are different. So each and every living body has to prepare their own proteins, and to prepare the proteins they need nitrogen. But the nitrogen which is present in the atmosphere is directly of no use to all these organisms. That nitrogen, it has to get converted into nitrites and nitrates and go into the soil. From the soil it goes to the plants, and from the plants it go to the animals. Okay, so that is a food chain which we have studied in chapter 7. Now we will come back to Rhizobium. What does Rhizobium do? Rhizobium stays in these nodules and they convert atmospheric nitrogen into nitrites and nitrates. Okay, so that is the use of Rhizobium to the plant. What do the plant do? What does the plant do? What does the plant give to the Rhizobium? The plant provides the Rhizobium with carbohydrates to live. Okay, carbohydrates to live. The plants provide the Rhizobium with carbohydrates. Okay, so there is a mutual relationship, there is a give and take relationship. That relationship is called as symbiosis. So that is said to be symbiosis. It is given in the next paragraph. Rhizobia living in root nodules supply nitrates, nitrites and amino acid to that plant, and in exchange get energy in the form of carbohydrates from it. Such a mutually beneficial relationship is called as symbiosis. You may underline that, it is asked. Okay, now you may see figure 8.2. Rhizobium is rod shaped. It lives inside the nodules. You may see bacteroid. Okay, bacteroid, that is the growth of bacteria inside the nodules of the plant. The diagram is not asked generally. Now the last paragraph, rhizobia produce nitrogenous compounds from atmospheric nitrogen. However, for this process of nitrogen fixation, they need leguminous plants like beans, sweet pea, soybean, etc. as host. Host means the plant's body on which they live, that is said to be host. Beans and pulses are rich in proteins due to nitrogenous compounds made available by rhizobia. Now you may write a question there, the examples of leguminous plants. What are the examples of leguminous plants? Beans, sweet pea, soybean. Beans, sweet pea, soybean. They are leguminous plants and they are rich in proteins. Why? Due to the nitrogen fixing rhizobium which live in the nodules. Now we don't eat the roots of methi. We eat the leaves of methi, isn't it? We don't eat the nodules. The nodules only provide the proteins to the plants, that's all. And the plants get proteins and those proteins we get. Okay. Now the next page, page number 90. After harvesting a leguminous crop, the leftover roots and other plant parts are deliberately dumped in farm soil to maintain its bacterial population. Now you might have learnt about crop rotation. Crop rotation is a method followed by farmers. For example, one batch of fenugreek, one batch of methi in a particular field, methi has nodules. After the methi is harvested, the roots of those plants are deliberately, means purposely, muddamun, they're deliberately dumped into that field. Why? Because there is rhizobia in it and it will help in nitrogen fixation, and the next batch will be some other crop which do not have nodules. Okay, but there are already the nodules are present in the soil. It could help in nitrogen fixation. The use of rhizobium has helped to reduce the use of chemical fertilizers and thereby their adverse effects. It has also helped to reduce expenses on fertilizers and thus works to the benefit of farmers. So what is the importance of rhizobium? Role and importance of rhizobium. It is beans and pulses, that is leguminous crops, are rich in proteins, okay, due to rhizobium. They are deliberately dumped in fields to maintain the fertilizer content of the soil. It reduces the use of chemical fertilizers and reduces the expense of chemical fertilizers. Now the last topic is rhizobial inoculation. It is a 2-mark question in a box towards the right hand side. Very important part. Nowadays, seeds are coated with rhizobial solution or powder before sowing. After sowing, rhizobia enter the plantlets. This is called rhizobial inoculation. So what is done? The seeds are taken. Okay, the seeds of the crops which are not leguminous. Okay, the seeds of the crops which are not leguminous. The seeds of those crops are taken and it is mixed and it is coated. That means it is put into a solution which contains rhizobia and then it is dried. Okay, and when it is sown in the fields, that rhizobium grows in the soil. Okay, so that is rhizobial inoculation. I will read out again. Nowadays, seeds are coated with rhizobial solution or powder before sowing. After sowing, rhizobia enter the plantlets. This is called rhizobial inoculation. This experiment has helped in the supply of nitrogen to cereal and other crops besides leguminous crops. Leguminous crops, they can automatically grow nodules and they get rhizobium and other nitrogen fixing microorganisms. Okay, so this is about the two useful microorganisms, that is Lactobacilli and Rhizobium. Okay, now I'll give you the homework. So this is the homework for today. How is yogurt made from milk? The paragraph you have to write. Write the uses of Lactobacilli. There are 5 uses given. Any 4 you may write. What is meant by symbiosis of Rhizobium? That paragraph, second last paragraph on page number 89 you may write. What is rhizobial inoculation? Page number 90, that box. Okay, this much for today. You may read it and write the parts and mark the parts which I have told you in the textbook. Okay, thank you.